Good morning, everybody. We're going to get started. I want to thank you for joining us this morning for the Publications 101 Symposium, How to Get Your Work Published and Avoiding Ethical Minefields. I'm Kurt Sigmund. I'm chair of the APS Publications Committee, and I'll be joined this morning by Dr. Kim Barrett, who is the past president of the American Physiological Society, a former chair of this committee herself, and a former editor-in-chief of AJP GI. Cell, sorry. I've said that twice now. You didn't correct me the first time. Sorry about that. I'm also joined by Rita Sheeman, who's the APS Director of Publications and the Executive Editor of all the publications of the American Physiological Society, and Christina Bennett, who is the APS Publications Ethics Manager for the Society. We're also joined currently by one editor-in-chief. Josephine Adams is the new editor of AJP Cell Physiology, and shortly Bill Yates, who is the editor-in-chief of Journal of Neurophysiology. And there'll be an opportunity at the end of the symposium to ask some questions of our editors-in-chief, so you could start thinking about what you might want to ask them now. And I know they're very interested in a back-and-forth interaction with you. So the outline of the presentations this morning are going to be, I do have a pointer. Here we go, excellent. I'm going to talk a little bit about preparing your work for publication in an APS journal, but of course a lot of what I'm talking about here is true of any journal that you choose to submit your scientific work to. We'll talk about choosing a journal, authorship and peer review, and copyright issues. Dr. Barrett will talk about the first issues and some of the minefields that come up in terms of plagiarism and data duplication. Christina Bennett will come up and talk about the yeses and nos of image manipulation, how to deal with blots and things like that. And then we'll have our question. We'll have a couple of scenarios to discuss in a group format, and then we'll finish up with the question and answer period. So let me start off with preparing your work for publication. In the journal, we'll talk a little bit about the overall process, some of the issues to consider both before you start to write and also during the writing process with regard to authorship and journal selection. Talk a little bit about the elements of a journal article, the review and revision process, and tips for enhancing your chances of acceptance of your work. So I think a lot of this is going to be intuitive for you. Why is publication so important? Obviously, if you don't have any publications, then really no one knows that you're doing any work out there, and there's no way to report. You don't have a project really until you report your data into the scientific repository, and the scientific community has an opportunity to review that data and then to utilize your own data in the advancement often of their own research. So your results must be available for others, or it is as if it doesn't exist. So publication is very necessary for that. In a more pragmatic point, of course, for most academic institutions, no publication, no promotion. This is the main yardstick of productivity that is measured in nearly all academic institutions. Really two things that we always think about when we think about academic productivity. It's publications and acquisition of research funding in one form or another to perform that research. And then, of course, if you don't have any publications, you have a very difficult time regaining your funding because you have to answer that question all the time, what have you done for me lately? And so reviewers are often taking a look. In fact, it's often one of the first things that reviewers of grant applications do is to take a look at the person's biosketch or CV and take a look at their academic productivity. And that's true both at very early stages where you're looking at the promise of a new investigator to do great things, but also in the mid-career and senior levels where they're asking, how well have you done with the last bit of funding that you were provided? And really, research publications are the key to this, so it's vital for your success. So this is, many of you probably already have some experience in publishing, some may not. If you're a young graduate student in the audience, raise your hand. So there are a few. So if you've not yet submitted your first paper, raise your hand. So there are a few in the audience. So thank you. This is really designed for yours. So in a classic model of submission, review, revision, and submission, you are completing your research and then you're preparing a manuscript. Now, this particular order here is not always the way to go. You can often begin to draft your manuscript at the earliest stages. In fact, I always recommend that you draft the methods section of a manuscript very, very early. It's often those minute details that are forgotten first in something that you might have done a year ago and exactly how you did an experiment. I also recommend, as we begin to start seeing a story emerge from a number of pieces of data, start having people in my laboratory begin to draft the manuscript, because it's often in the narrative and the flow that helps you identify, well, you know, this would be better if I did this experiment and this experiment, or tie these two things together a little nicer. So in fact, this is a process that very successful people do continuously during the conduct of a project. Once a manuscript is completed, it's submitted to a journal. We'll talk about journal submissions in a few minutes. It's assigned to an editor-in-chief who will then assign it to often an associate editor. It's one of the senior editorial team that makes up the journal. Often that decision to assign it is based on the expertise of the associate editor, with reflection on the content of and the subject of your own paper. It's given out to a number of peer reviewers, and hopefully within 2 or 3 weeks those reviews come back and a decision is made. And then you have this decision tree here, adapted for some of our own journals. So in one case, in the best possible scenario, your paper is accepted right off the bat, okay? And I could tell you that that is probably a minuscule percent. In fact, I can't remember a time when a paper from my lab got accepted first time around, okay? More likely, if it's something the journal is interested in seeing again, it's going to be a decision to revise the paper based on the comments of the peer reviewers, okay? And so you would work with your advisors and your collaborators and your co-authors on responding to the reviewer criticisms. They could be just written criticisms. They could be a request to do additional experiments. However you decide to do that, you will then resubmit your paper. Basically, it will go back to the top again. It'll undergo re-review. And at this point, it'll either be accepted, rejected, or in some cases even revised again. And the last paper I just got accepted took 3 revisions before it was finally done. So these things, they often get a little faster at each step, so it's nothing to worry about, but this is not this is pretty normal. Of course, one possibility is that the paper is rejected. This happens to everybody. It's just part of the life cycle of science. We tend to aim high in all of our research, and that means we're going to get papers rejected, and that happens on a regular basis. Ultimately, we'll have our paper ultimately accepted and published. In the journals of the American Physiological Society, we actually have our regular tier journals, which you're going to hear about in a second. I have a list coming up. But we have another journal called Physiological Reports, which is utilizing the new open access model where you pay a fee for publication, and then the paper is there and freely available to anybody, anywhere, to review right away. On occasion, if a paper if there's a decision to reject the paper from one of our primary journals, sometimes there's a recommendation to refer that paper to Physiological Reports, and you have the choice on whether you're actually going to transfer. And we've built this in a very simple way so that you could transfer the paper basically automatically. You could respond to the reviewer concerns, and often that section of the peer review goes very, very quickly and hopefully leading to acceptance. Okay, so in terms of authorship, it's really important to decide on the authors, the order of their names on the paper as early as possible. If at all possible, do this really early in the project, even before starting a project. Of course, at that stage, you may not know who the authors are ultimately going to be, but it may be a collaboration. It may be a collaboration with you and another student that's working in the same laboratory or a neighboring laboratory. It is really best to sort these kind of issues out as early as possible. And in our experience, this is the type of thing that can harbor some ill feelings down the road if it's not handled appropriately. And this is when you're often dealing with your friends and colleagues, and the last thing you want to do is have an author dispute with them. So having this agreement with yourself, with your advisor, and having an understanding on how people are going to contribute to that science and be rewarded for that is really important. And then the other thing to remember is that authors should include only those who have made a substantive intellectual contribution to the project reported and can defend the data and conclusions publicly. And I think this is a really good standalone requisite for publication. So for the criteria for authorship, they should generate at least part of the intellectual content, conception or design of the work, data analysis, interpretation. Now keep in mind, intellectual content is not only the idea that generated it. It could be incremental ideas on moving from one experiment to another. And intellectual content can include methodology, okay? So even, a research assistant or a technician in your lab can be providing intellectual content by simply doing the experiments and then making recommendations for methodological changes or troubleshooting. That is intellectual, okay? It's not the overarching idea conception, but it is part of the intellectual content. Draft, critically review, or revise the intellectual contents of somebody who's involved in the drafting and reviewing of that manuscript. And of course, you always have to approve the final version to be submitted. Every author must approve the final version as it stands before it's submitted. And it's something I take a lot of care of. And Michael's in the room. He's been a co-author in a number of papers from my lab, and he knows that at the end I send out an email and I don't submit it until I get an actual statement back. This is ready for submission, okay? And so that's really important. That way everybody agrees with its submission. So all three criteria should be satisfied. So choosing the right journal, who's the target audience, really it's what you're asking for is who's going to read the paper. So who is your target audience for the data set that you are submitting for publication? If it's a specialized area, so in my laboratory it would be blood pressure regulation. We would be submitting it most likely to a journal which focuses on blood pressure regulation, as opposed to I wouldn't be submitting it to a journal that focuses on respiration. That doesn't make much sense. It's not going to have the right expertise in review, and it's not going to have the right readership going to that journal to see our content. So this is important for choosing the right journal. addressing the import and significance of the findings. This is very helpful to seek input from your colleagues. You're often going to be doing that, especially as trainees in a variety of oral presentations at your institution. And it's just very helpful to try to decide on the journal before you begin to write, because so many journals have different formats. And the format can often influence the flow of information. Is it a journal that puts the methods up front or a journal that puts the methods at the end? It does influence the way the information flows in the paper. So this is the series of APS journals. It's a fairly large portfolio. There are the 7 American journals of physiology here on the left, and then 5 additional scientific journals that go by a different name. Here's Physiological Reports, which I refer to in a minute, and I'll come back to in a few minutes. There are 3 review journals in the APS. These are ones that publish review articles which summarize the literature on a topic. Physiology tends to short, focused reviews. Physiological reviews are very, very long, comprehensive reviews on a topic, often with hundreds, if not 1,000 references associated with that. And comprehensive physiology is a growing journal in the portfolio. So the essential elements of a manuscript are the introduction, the methods, the results, and the discussion. Also, there's an abstract which summarizes, often in 250 words, just the bullet points of the study. The introduction is designed so that you can explain to the reader what was known, what is unknown, and then why you did the study. So you're basically asking the question, why? The methods is answering the question, how? How did you do the study? What methods did you employ in performing the study? And this is probably the most important part of a paper, but is often given the least attention. Okay, and this is something that's very important. The journals of the APS have no word limit and no page limit, and so the method section can be as detailed as you want it to be. That often doesn't mean numbers of microliters to that level of detail, but really giving a comprehensive view on how you did it and ensuring that your work, if you're citing work of others, is cited correctly. The results section is the what. What did you find when you did these experiments? The discussion is, now that I have these results, what does it mean in the context of the broader state of knowledge on that particular topic? And so all of these are critical parts of the paper, and each of these will have some commentary from peer reviewers as you're getting your papers back. So some tips for success. It's really helpful to know about the journal before you submit, that you've looked through a number of tables of contents. Hopefully this is a journal that you're reading on a regular basis. It's helpful to know not know personally the editor, but something about what the editor studies. It's also very helpful to learn a little bit about the associate editors and what their area of expertise is, because when you submit, you're going to be given an opportunity to choose your preferred associate editor. So if there's somebody who has expertise directly in your area, they're the ones that you really want to suggest in terms of reviewing or handling the review of the paper. Read the instructions to authors. I can't tell you how often this becomes an issue. Often people don't simply read the instructions and they make careless errors. Avoid carelessness. Take a look at the spelling. Trust your spell checker, but you have to train it for scientific language, don't forget, often is the case. Look at the grammar. Make sure other people, if English is not your primary language, get somebody who's a native English speaker to review the paper. for you. This is part of the professionalism in publishing a paper. If the paper looks professionally done, it's going to be elevated just a little bit in terms of how it's going to be perceived. Make sure the references are appropriate and accurate, okay? This has happened to everybody or will happen to everybody. You go in a method, you pull out the citation for that method, and you realize that, in fact, that paper is citing the method. And then you go to that paper, which is citing the method. And then you go to that paper. And it takes you 10 papers, 10 generations of papers to actually find the method that you were doing. So please cite your work appropriately. Cite the primary data in support of something that you're making a statement about, whether it's in the introduction or in the discussion. There is nothing wrong with citing a comprehensive review and saying reviewed in, okay? If it's something that you just want to highlight that this is an important topic that's been reviewed comprehensively, nothing wrong with that. But typically don't cite a review to make a point about a primary piece of data that was cited in an original paper. Make sure that you have all the correct file formats. It's going to be particularly important for figures. Remember that, and you'll see this in a minute, the paper, when it's finally accepted to one of our journals, will appear online first as that final PDF, okay, which is the way you submitted it. So you want to make sure that you're comfortable with people reading and looking at the paper in the way you submitted it. So that goes towards his professionalism. If you know there's a mistake, don't tell yourself, oh, I'm going to wait until I get the galley proof to correct it, because that is published already. It is one of the published versions. So if you know there's an error, go back and have it corrected. So the website for instructions for preparation of manuscripts to the APS journals is right over here. There's a wealth of information on all aspects of publication. If a paper comes back for revision, that's a good thing, okay? Like I said, very, very few are accepted right out. So you're in great company if the paper is, we would really like to see it again, we're inviting you to revise it, and we'll think about it. We'll think about accepting it if you can respond appropriately to the reviewer comments. Okay, that's a great decision letter. It's okay if, when the reviews come back, you get a little irritated by something that a reviewer said. This will happen more often than not. Scream, yell, but then chill. Okay, and get it out. But don't put any of that emotionalism back into the revision when you're actually ready to write it, okay? I could tell you that some of the best papers we've published have been ones that have either been brutally rejected from another journal or have had really stringent reviews. And after we're irritated and we scream and dance around a little bit, we read through it and say, you know, they're right. This can be better. And there's a couple of instances where we've gotten decisions back and we've really realized that we told the story wrong. Okay? And when you didn't realize it, you get tunnel vision sometimes on your own work, right? And you didn't realize it going in. But after the review come back, you realize you tell the story wrong. We rip it apart, we start it again, we reorganize the figures, and there are a number of occasions where the paper turned out to be a much, much better paper for the effort. So it's really very helpful. Look for clues from the editor. An editor or an associate editor in their letter to you or in a comment section by the reviewers will give some helpful hints on the things that they deem are the most important to address. And don't hesitate, if necessary, to get clarification from the editor, associate editor, regarding a point, okay? You could write back, do it after you've calmed down, and you know, the text of the letter, you go with your advisor and the co-authors, make sure it's in the tone that you want it to be. But often they can be very, very helpful in doing that. So responding to the reviewers, you may have to complete additional experiments as needed. You want to address all comments in a point-by-point fashion. What I always do is I actually take the whole review verbatim, I cut it and paste it into a Word document, and I label it comment 1, comment 2, comment 3, and then it's response 1, response 2, response 3. And we start composing the responses as we go along. We edit the manuscript and deal through. So on some responses you might leave blank for a while because you may need extra data to incorporate. Resist the temptation to give up impassioned response to review. Keep emotionalism completely out of it. Just deal with the facts. If you really feel strongly about a point and the reviewer is wrong, you can stand firm. Do it diplomatically. It takes some experience to write those responses. It's very helpful to get the advice of colleagues. I still get the advice of my colleagues when I feel that I'm in the right, a reviewer is in the wrong, and I want to write it in just the right way. Okay, and my colleagues often help and change my verbiage a little bit, and it often gets better. And the reviewers will often respect your positions, especially if you responded positively to other things that they were suggesting. Thank the editor and the reviewers for helping you. No matter what, they have invested a lot of their time, their free time, their unpaid effort time to help you review your work. They're helping you make your own paper better, even though it sometimes doesn't seem that way, but that is what's actually happening. So thank them for that. And again, get a neutral colleague for a response. In terms of major reasons for rejection, the simplest one is that you simply submitted it to the wrong journal. Okay? It's inappropriate for what they think the focus of their journal actually is. Perhaps you've aimed really, really high in some of the highest top-tier journals, and it wasn't considered to be of significant general interest to the scientific community. And that happens pretty often. It can also be rejected if the editors feel or the reviewers feel it's simply confirmatory or incremental of other results that are out there. And that's really where it's important to understand the literature surrounding your own area to make sure that you're simply not repeating somebody else's work, and you're not making a small increment, but you're really extending the science away. So avoid the LPU. What's an LPU? Does anybody know? That's right, the least publishable unit. Okay, so little mini paper with just a figure or two is often considered. It's the first thing that raises a flag, you know, to an editor and the review team. If your study is poorly designed or it's actually could be overly inclusive, you may have to either focus or become more broad. Again, professionalism is really important here. If it's poorly written, so it could be great science with the greatest data on the planet, but if it's in an ugly package and it looks messy and haphazard, it doesn't come off as being professional. And you don't want this to happen, because if someone feels that you're sloppy in presentation, they might feel that you're sloppy in experimentation at the same time. So it's really important to have this polished up beforehand. So in summary, do the study with the paper in mind, assign authorship appropriately, choose the right journal, seek input from your colleagues, remember who the reviewers might be, and don't try to guess who the reviewers are, okay? I've been an editor for a long time. Everybody guesses wrong, okay? Always. Okay, 99.999% of the time, you think you know. No, it's completely wrong. So it's something to keep in mind. And if you're unsure about an ethical issue, ask about it. Ask your advisor, ask others in the laboratory, ask your colleagues to help you with that. So just a few things in closing before I hand this over. The APS articles are published in 3 forms. The article in press is the pre-edited PDF. It's that final version that's assembled, and it has the same type as, you know, the Word document, and the figures are at the end. So that's the first thing that comes up. It's usually within a week or 2 of final acceptance. You have the final journal issue that looks like it's beautifully typefaced and put onto the journal format that's online. And then for a print journal, it's the final print issue if it's actually still in paper. I mentioned a little earlier physiological reports. This is an open access journal that is a partnership between the American Physiological Society and the Physiological Society in the United Kingdom. It started in March of 2013. We've now had a full year of submissions. It has a slightly different model. Instead of paying for page charges after the fact, you actually pay a fee on acceptance. It's a one-time fee. It doesn't matter if the article is 1 page or 20 pages. There are 2 ways to submit to that journal. The normal way, where you have a paper, you decide this is the place you want to put it, or it can, what we call, cascade from other journals. So if it doesn't quite meet the bar for publication in, say, one of the AJPs, it may be suggested to you that you send it to physiological reports. The papers are reviewed in a different manner. So most journals review with scientific rigor and an advancement in the field. PR is based solely on the scientific rigor, technical and ethical standards, and that the study is well designed, but it doesn't necessarily have to be a major leap forward, okay, in the understanding of the field, and it does actually include publishing negative data, okay, which is something we all generate in too much abundance often, okay. The editor is Sue Ray, and the deputy editor is Tom Clayman, and this is the information. You can go to this website for that. I think this might be my final slide. There's a lot of verbiage on it. It's something simply to remember is that when you're publishing in our or any other journal, you're basically assigning the copyright of that work to the society or to the journal. There may be times when you're giving an oral presentation, when you're writing a review article, when you're writing your thesis, where it's very convenient to actually use a figure from somebody else's paper or even one of your own. You know, there might be a nice schematic. You do need to obtain permission to do that. And it's not usually from the author. It's actually from the copyright holder. And so you go to their instructions to authors, you'll often see something about requesting permission. And it's become very easy, and in fact, there are some clearance centers that allow that to be done much easier. So with that said, I probably have rambled on too long as it is. I'm asking Kim Barrett to come to the stage and talk about Ethical Minefields Step 1.
So I'm going to talk about some of the things that you want not to do when publishing your work. And I'm going to talk about 2 aspects of this, avoiding plagiarism and data duplication. And the sort of genesis of this session is really that nobody comes out of the womb knowing everything about publishing a scientific paper. And when you're early in your career, things that will seem obvious later on really are fairly obscure. So why do we care about this? So there have been quite a number of high-profile cases of problems with scientific publication. So this is a Japanese scientist called Dr. Obokata, and just in the last year or so, basically admitted to problems with the science that she published. There were duplications of images. There were images of one cell that really wasn't what she said it was. And there has been question about the whole technique that she published, which was basically saying that you could reprogram stem cells by briefly exposing them to acid. Now, she's really essentially lost her career, but her supervisor at the Riken Institute in Japan, who was the corresponding author of her articles that were retracted, actually committed suicide. So that's a very sad ending. Here's another stem cell guy. He also basically fabricated the work in his stem cell papers. And the interesting thing about him was he had a co-author from the United States who was very happy to be on these high-profile papers that were published in Nature. But when they were retracted, he turned around and said, well, I really didn't know anything about this. I was just on the paper as an honorary author. So that's really gets back to what Kurt was saying about authorship. Don't take authorship of a paper if you don't, if you're not willing to take responsibility for the data. So this is Dr. Huang. And then finally, this is an investigator called Eric Pohlmann. He has the distinction of being, I think at this point, still the only author that we've ever banned from submitting to the APS journals. As it says here from this news item, he fabricated data in 17 grant applications to make his work seem more promising. At the outset, it helped him get $3 million in government funding, but it didn't do him very good in the long run, and I don't think he really cares anymore whether he can publish papers, because he was actually sent to jail for this fraud. So these are the really extreme cases, but you don't want to be in this rogues gallery. So being a scientist is very much an enterprise based on trust. We expect each other to be honest. We expect when you read something in the literature that it was done in an intellectually honest manner. It's important that we accurately assign credit. As Kurt was saying at the beginning of his presentation, papers are really the coin of the realm. They're the thing with which we judge people's productivity. It's the thing that's going to get you promoted. It's the thing that's going to get you grant funding. And so it's important that we accurately assign credit to the people who actually did the work. We expect people to be fair in peer review, because it is review by peers. Do unto others as you would have them do to you. We expect people to be collegial, to have transparency if they have any conflicts that might bias their approach to peer review, for example. And particularly important for us as physiologists, a lot of our work involves human and or animal subjects, so we need to protect those subjects in what we do. Sometimes this goes awry. We are not the only publisher of scientific journals to have the experience that misconduct has been increasing among submission to our journals. I'm glad to say that recently we've seen that plateau and maybe even go on a downward trend, and we're very pleased to see that. And that's part of the reason why we have these sessions, is to sort of try and educate people as to what is and is not permissible. The vast majority of scientists are entirely ethical, but if you survey people anonymously, as was done in this Nature paper cited here, people will admit that every once in a while they'll do something that's a little bit questionable. So it is a fact that sometimes people cut corners, and there are risk factors that lead people to think about cutting corners or just not doing the right thing. The first and most important from our perspective is that people may be inexperienced, or they may lack mentoring on what are the right way to handle things, whether it's handling the reuse of data or how to handle figures, as Christina will talk about in a moment. It's also a risk if there are multiple authors on a paper, and increasingly as people are working across the boundaries of disciplines, you can't possibly know absolutely all the nitty-gritty details of every piece of data in the paper. But as an author of that paper, it is your responsibility to try and figure out what the data from your collaborator really means and how it was generated, and wherever possible to actually look at the raw data and satisfy yourself that you understand where it came from. Because as the number of authors on a paper increases, that does increase the risk for problems with the paper. It's also the case now that we have a lot more tools at our disposal that can help us put figures together, but equally they can be used inappropriately to manipulate figures in a way that doesn't allow them to truly represent what you saw in the lab, and Christina will talk about that. And then there certainly is competition for jobs, competition to get grants, and perhaps if there's increased competition, people get very stressed and they try to really get any advantage that they can. But I think of these four possible factors for ethical violations, it's the inexperience that is A, the one that we think is probably the most important, and B, the one that we can actually do something about. So we take ethical matters very seriously at the APS, as do the vast majority of publishers. And if you go on our APS website under the publications section, you'll see our ethical policies that are quite clearly stated. If you violate the policies, things can happen to you. One of the things that can happen is that we can sanction people if they are guilty of the most serious types of offenses. We can notify the author's institution, which is kind of a world of hurt, because then they really have to start digging into everything about your publication to figure out what happened. So it could potentially be a career breaker. And so what I want to accomplish over the next few minutes, as does Christina, is really to give you some information that allows you to avoid these pitfalls. There's also an APS professional skills training course if you wanted to delve into this more deeply. So what are some of the things that happen? By far the most common thing that we see at the APS is manipulation of figures that is not appropriate, that's not in accordance with standards, but it's not fraudulent either. It's not like people are trying to do something wrong, conceal data, show data in a way that actually doesn't represent reality. But it is nevertheless inappropriate, and Christina will talk about that at length. We see duplication of data from one publication to another, actual duplicate publication where people try to publish the same paper twice. That's a great way to bump up your CV, right? If you can publish the same paper in many different places, obviously that's not appropriate. Again, as a physiological society, we're very concerned that our animal studies that are reported in our journals are done in accordance with the highest standards, so we see concerns with animal welfare. Rarely, we see more significant issues such as actual fabrication or falsification of data. I'm from the University of California, San Diego, where we had, when I was a junior faculty member there was an individual called Slutsky, which I thought was a great name, and he was basically submitting papers every week. And those of you who are working at the bench know it takes a long time to generate the data that you need for paper. And finally, the institution said, wow, he's sending in a manuscript a week. How is he doing this? Well, he was just making the data up. It's very easy to generate manuscripts if you don't actually have to do the experiments. So eventually he was caught. But also data fabrication can include things that are a little more subtle, like inappropriate and actual fraudulent manipulation of figures, which Christina will also speak to. Plagiarism, and we'll go into more detail what that actually means, but basically misappropriating other people's work. We see human welfare concerns. We see disputes between authors. So Kurt's comments about figuring out who the authors are, the sooner that you do that, the better off you are, because that means you're less likely to get into an argument about it later. We occasionally see conflicts of interest, people reviewing who perhaps should not be reviewing, people who report findings that have the potential to be colored by commercial interactions and so forth. And then rarely we see things like reviewers behaving badly, issues with submissions, people sending in papers that the other authors haven't seen, and so on. So I'm going to focus my remarks on duplication of data and duplicate publication and plagiarism. So what do we consider to be duplicate publication, data duplication, or you'll also see this referred to as either redundant or prior publication? So we expect original articles to be original. That means that they don't contain data from other articles, they don't contain text that have been published in other articles, and they don't include either tables or figures that have been published in other articles, including your own. So anything that has appeared in any prior publication should not reappear in an original article. Now, of course, different rules apply if you're writing a review article. You might want to reuse some data. You might want to reuse a figure that's already been published, and as Kurt alluded to, you can get permission to do that. But for an original article, the material should be original. So redundant or duplicate publication includes using something that you've already published, or at least usually using your own materials. And it's also referred to as either auto-plagiarism or self-plagiarism. So how can you avoid doing this? So if we're talking about data, don't include material from a previous study in a new one, even if it's for the purposes of statistical analysis. So you really want to make the paper original. So in many cases, that requires you to repeat control groups as needed. Now, it is the case that not only is this ethical good practice, but it's scientific good practice. You want to repeat your controls for the experiments. But a sort of perhaps more benign form of this is when you cut and paste text from one manuscript to another. So I've been in the business kind of a long time. I predate word processors. I remember when I used to write all my manuscripts out longhand. When you do that, you're much less likely to copy things. But these days, it's very easy to start from the paper that you just published and take the whole method section and paste it into the new manuscript. I really urge you not to do that. First of all, because it is ethically inappropriate. At a minimum, you might be violating the copyright from the paper that you just published by reusing that text verbatim. The second reason, and actually the more important reason, is people often get things wrong when they cut and paste into a new manuscript. For example, the previous study that you did might have used rats. The new study uses mice, and your methods start talking about rats, and the reviewers get very confused. And you also often change your protocols over time, or there's important changes in the way that you've designed the experiments. And if you copy and paste, you might miss those. So, and finally, we're all under pressure for space in journals, and if you've already published a detailed description of a method, you really don't need to publish that again. You can just cite it. So don't cut and paste is the short answer. So what are the exceptions to these rules? So very occasionally, you might want to reuse some data. So an example would be if you have a set of human subjects that you're doing a set of distinct research questions, but the baseline data that of those subjects that you're studying, for example, their age, their sex, their baseline blood pressure, for example, that might be the same for every study, and it might be important to have those baseline data for every paper so that you can understand the context of the actual experimental data. Another example would be highly invasive studies in animals where you can't justify repeating the controls over and over and over again. And in fact, if you do animal work, you'll find that when you apply for an animal protocol, you will be asked to say, what are you going to do to reduce the number of animals that you're using? So there are certainly justifiable sometimes to reuse some control data from animal studies. And then a third example of when you might be able to reuse data in an original article is if you need to compare your new data with something that's already been published. And typically that would be if you need to graph your new data on the same graph with either your data or somebody else's previously published data. So how can you avoid then, if you need to do any of these types of things, being accused of duplicate publication? The very first thing you need to do is tell the editor. In Figure 5, we have graphed not only our original data, but the data that we published in American Journal of Physiology last year, and give the citation. And you should tell the editor in the cover letter, but it also needs to be in the paper itself with citation. So before you even consider duplicating data from another source, make sure you're very, very explicit in disclosing it. Duplicate publication. So this is basically submitting or actually succeeding in publishing the same paper or substantial parts of a paper in more than one place. So this is kind of thematically related to what I've just been talking about. That was more focused on taking a piece of data and putting it in multiple places. This is kind of wholesale, the whole paper. And certainly we have seen over time people who have attempted to publish a paper that's already been published in one of our journals. Now, when I was starting out, I thought it was like, you know, writing a short story or a book. You want to send it to a whole bunch of publishers, and hopefully one of them will say yes. And it would save us all a lot of time if we could take our paper and just send it to 5 or 6 journals and see who took it first. The reason that we don't permit that is because, as Kurt has already alluded to, people put in a lot of time reviewing manuscripts, serving as an editor, and they want to know that the work that they're putting in is actually going to be worthwhile. So you cannot submit your paper to more than one journal at a time. So pick your first journal, send it there. If they like it, great. If they don't like it, go on to journal number 2. So wait until your paper is rejected before you submit it elsewhere. And then the other thing that comes up quite often is if your paper is not rejected, but you're invited to revise it and resubmit it, you may decide not to do that. You may decide that you can't do the experiments that the reviewers are asking for, or the first author on the paper has finished their PhD and they've gone off to do a postdoc and they're no longer available to do the experiments. You don't have the reagents anymore. The first author has dropped out of science. For whatever reason, if you're invited to revise and resubmit, you may not want to do that. You may choose not to do that. If that's your decision, let the editor know. And the reason for that is if you then decide to just resubmit somewhere else without revising the paper, you may very well get the same reviewers who see the paper again. And they will write to the editor and say, hey, wait a minute, this paper is still being considered by the first journal. Why are they submitting it to a new journal? So if you are not going to resubmit, withdraw it from the first journal. So why does this matter? Why does any of this duplicate stuff matter? As I said at the beginning, original articles are expected to be original. And if the same data starts getting published in more than one place, it kind of gives a false impression of the reproducibility of a finding, of the impact of the finding. If you're working in the clinical arena, often clinical studies are aggregated into meta-analyses to try and get an understanding of the true clinical efficacy of a treatment or an approach to a disease by aggregating lots of smaller studies. If the same data is being published in more than one place, then it makes it look like there's more evidence for a treatment than there really is. And then, of course, the other ethical dimension of this is you don't want to inflate your true productivity. And then plagiarism. What's that all about? So the definition of plagiarism is taking the work of another, copying somebody's figure, table, or even just the words from an unpublished or a published paper without attribution. You can avoid this by providing citation to the work of others, very important, we all know that. If you are writing a review article and you want to reuse a figure, you need to get copyright permission. And you should avoid just using the exact words from another source, even if you give a citation, unless you put it in quotation marks. So if you find that somebody has said something so beautifully that you can't possibly think of another way to say it, make it a quote and say, Dr. Sigmund in his paper last year said, quote, and whatever it is that he actually said. So make it very explicit. It's not enough simply to write the sentence and put a citation at the end, because that makes it look like you came up with that sentence and the idea maybe was in Dr. Sigmund's prior paper. Plagiarism is particularly an issue in review articles, and many of you are at the beginning of your careers and haven't yet published your first research paper, let alone sitting down to write a review. But when you do start writing review articles, it's very easy to plagiarize things, or more to the point, as you start writing more and more review articles, they're at risk for self-plagiarism because you just are using the same thing that you have talked about before. You get asked to write the 10th review on the subject matter that you're working on, and you can't think of any new ways to say things, and in fact, maybe you cut and paste the introduction to the review from something that you've written before. So the APS, like other publishers, is concerned about this, and so in fact, we're now screening our review article submissions with software designed to pick up plagiarism. And if we find that, we go back to the authors and say, we think you have a little bit of a problem with this. And another journal that I'm working on now, the Journal of Physiology, that the PhysSoc publishes in the UK, they screen all of their articles with plagiarism software. It's just done automatically as soon as the paper is submitted, so even original articles. If you're writing a review article and you take notes on the papers that you've looked at to familiarize yourself with the field, either rephrase them when you take the notes or make sure that you clearly put these in your notes as direct quotations. So if it is a quotation, make sure that you label it as such, because when you go back to put the thing together, you may forget that you actually wrote down the author's exact wording. But the bottom line for all of this is there's always another way to say things, and you want that work to be original to you. So why does this matter? Well, first and foremost, of course, is intellectually dishonest. In essence, you are stealing somebody else's ideas and somebody else's way of saying things. But it's also legally wrong because papers that are published, review articles that are published, most of the time somebody owns the copyright to that work, and even if it's your own work and you reuse it, you are violating the copyright holder. So, and that's then a problem for the second journal. They might be subject to an action by the copyright holder. So in conclusion, for my part of this, we do know that science is an enterprise where we have to trust each other. You know, when you're in the lab, your PI trusts you to tell them exactly what you did and to be completely truthful in talking about your data. It's the same thing in the wider scientific community. We all have to trust each other because we build upon each other's work. So you have to be confident that the original work is sound and is not in any way ethically compromised. So when we have ethical lapses, even if they're totally unintentional or if they are due to the fact that you simply don't know what is or is not appropriate, it really betrays the trust that we need to have in each other for science to move forward. And of course, if you do something really dire, that may have a huge impact on your career, including perhaps ending it altogether. So it's important that we all understand what are the ethical standards that govern publishing, and if you don't know something, whether something is appropriate or not, please just ask. Ask your PI, ask your colleagues, talk about things, because by doing that we can all make our publications stronger. Okay, and so now I'm going to pass this off to Christina Bennett, who is our Publications Ethics Manager at the APS, and Christina's also bringing her yet-to-be-born daughter up for her first scientific presentation.
Thank you, Kim. As Kim mentioned, one of the main issues that we see with our authors has been image manipulation. And for one reason, the APS art editors now screen the digital images or the photographs in all accepted manuscripts for image manipulation. And what they look for is that the submitted materials faithfully represent what was originally captured. And so this is an example where an image was submitted to the journal. This is a Western blot. It was a loading control for protein expression. There are some variables sitting on top of it. And what the art editors, from here, it looks pretty clean and a nice, well-loaded blot. But what the art editors can see with their software was that there was a line going through, a line in between lanes 3 and 4, which you probably can't see. And they also could detect that the lanes 3 and lanes 4 seem to have a more enhanced coloring of the samples. And so we asked the authors for the original captures. And what we found was, indeed, there was a line between lanes 3 and 4 because they were on 2 different blots. But what we also saw was that lanes 3 and 4 were well enhanced to make this loading control look more even. And so if you think about it, this submitted material does not at all represent what was originally captured. We all clear on that? Okay. So this article was not published because what the reviewers looked at to analyze whether this paper is worthy of publication does not represent what was actually performed in the laboratory. And if you think about all those variables that they looked at as changes due to that experimental procedure, if the loading control wasn't well loaded, then all of that data sitting on top of it is not true. So APS has a set of image manipulation guidelines that many journals now follow. They are kind of a list of don'ts, but I'll go through them quickly here and then give you some examples for the rest of the presentation. So authors should not move or remove or introduce or enhance any specific feature within an image. Authors shouldn't adjust contrast or brightness or color balance unless it's applied to the entire figure. If gels or blots are grouped or rearranged, authors should declare that in the image itself and within the figure legend. Authors shouldn't quantitatively compare data across different experiments unless it's justified within the text and within the legends. And if the same image is presented in multiple panels, often this might be a loading control or a control photograph, mention that in the legend, because if it's not explicitly clear that you're doing the exact same experiment and presenting 2 different portions of it, it may look like you're reusing the same loading control over and over again, just because it looks, looks okay, but it's not for the correct experiment. Okay, so I will say, I want to do a disclaimer. We all manipulate images, and it's all okay. We all have to take an original capture. We all crop it in some way to make it smaller because you just want to focus on the important information. We all adjust contrast in some way to make the main piece of information stand out a little bit more. We resize it, and we present a complete figure for our manuscript. But these same types of adjustments can get overzealous. Sometimes we'll see excessive cropping, missing backgrounds, resizing and reshaping, and then the submitted figure looks slightly different from what was originally captured. And so throughout this presentation, we just want to encourage you to make sure that what's submitted really holds true to what was originally captured. Okay, and what we see a lot of is undeclared removal of lanes or undeclared rearrangements within the figures that are submitted to the journal. And what we'll see is that the art editors, when you see it, you might not catch this, but the art editors can detect if there's a cut between multiple lanes. And so we'll see a figure that looks quite clean, and the art editors will detect that there's a line missing in between the second to last, or the missing, that there's a line in between the last sample. And so what we encourage is if you are going to remove a lane because it's an unnecessary sample, that you add a space or a line and add a note in the legend that said that a sample was removed because it was unnecessary. Another example of what we've seen is composite images. Again, this is staining of cells. The cells' nuclei are stained blue here. And when the art editors went to prepare this figure and analyze it for image manipulation, they found that it was in multiple pieces. And again, we queried the authors as to why they put all these pieces on top to make it look like one capture. And they said, well, we just didn't get enough cells within any photograph, and we really wanted to make it look more clear as to what we saw within the experiment. So we encouraged them to now box it because we really need to make sure that the readers are aware that this didn't come from one photograph, and that when they're trying to repeat the experiment, they may not get as robust a signal because the original authors didn't get that robust of a response. Another thing that's actually increased in the past couple of years is removal of original background. We'll see submitted figures that are here on these beautifully clean backgrounds. They're either all gray or all black, and the bands stand out beautifully. But there's no detectable information for the art editors to know where these bands came from. Are they from one gel? Are they from multiple gels? Have things been rearranged? Right? You can't really tell. This is another example. I call them floating bands. Clean as can be, right? The bands are clean. But there's no information to say where these samples came from. And so what we do is we just ask authors to restore the original background. Now, this may be dirtier, but at least you know that all the samples were loaded in the way that they were supposed to be loaded. They're presented in the way that the experiment was run, and you know that, okay? The other caution I have when things are removed or rearranged is that once you put things in pieces, you might adjust contrast for one sample differently than you adjust the contrast or brightness for the rest of the samples that you're connecting it to. And so the more you edit and the more things get enhanced, it's hard to maintain consistency across the entire image. And other times we find that the same image appears in more than one place within the figure set, and we question the authors, and they say, oh, it was by mistake. We cut the figure, and we inserted the wrong figure in multiple places. And so by keeping the samples together and presenting it as the experiment was performed, you really avoid these types of situations. Another example that our art editors found was a tissue section that was labeled, and when they went to analyze the image for manipulation, they found little white boxes covering over portions of the photograph. And when they removed the white boxes, they could see that debris, maybe staining debris, was being covered over by the boxes. And of course, we asked the authors why they did this, and the author said, we just wanted to make it look cleaner. But if your antibody staining or however the tissue is being experimented upon, if the experiment isn't as clean as it can be, you really need to tell the authors that it's not that clean. And this is an example from the Journal of Cell Biology where their art editors analyzed this piece of data that looks like there's punctate staining around some cell membranes. And it looks very clean, very pretty, very punctate staining here. But what they found was that all these little dots were enhanced. And the real image did not show as robust of staining. And so I'm not sure what the Journal of Cell Biology did, but clearly this is a much stronger result than what was actually captured. And it's really, as much as one wants to make their data look as best as it can be, one really has to present the data that was actually captured. One of my pet peeves is finding loading controls sitting on the bottom of a number of variables, and then each variable has its own reorganization pattern. And so obviously, if these samples weren't run in the same order as the loading control, then this cannot be the appropriate loading control for that specific experiment. So I just encourage authors to present the loading control for the specific variables that are presented. I realize that that may add an extra band or an extra lane or sample to the final figure, but it's important for readers to be able to compare how these changes in the variables are related to their specific loading control. And again, you can make a mention of why you're presenting maybe the same loading control twice by making a note in the legend that this is from 2 different experiments and that one had to be rearranged for final presentation. Okay, the last key word that I always kind of give pause to is the word representative data. And often it's when things are spliced or rearranged, because what I find is that representative means that it's been reorganized in some way. This is an example where the loading controls are in the bottom panels and the variables are sitting on top. And so this piece was actually in 2 pieces, and this piece was also in 2 pieces. And with our forensics tools, we could actually see that lanes 3 and 4 here are duplicated and pulled forward for it to become lanes 1 and 2. And so when we asked the authors why they did this, they said, well, we never got a fully clean experiment. We knew it would work, and this was the best one that they could show how representative the data would be if the experiment worked every time, right? So this really would not get published in our journals. We would not ask for a correction because that data never existed, and this falls along the lines of fabricating data, making it up. And as much as this is one example, one sample, one study within maybe 9 figures, if it's not true, then we can't publish it. So I do have a few tips. Please review journal image manipulation guidelines. We now all use Photoshop or some kind of photo enhancing software, and we all work really hard to make our data show and shine as well as it can be for journals. But make sure that what manipulations are done are according to the standards of the journal. Keep image adjustments to a minimum. When you take an original capture and put it in prepare it for a figure, manipulate it as little as possible, because if you work on this over months, every time you touch it and every time you clean it up, it gets a little bit different from what was originally captured. If there are major adjustments made on the figure that's outside our standards of cropping and resizing and adjusting contrast, declare those adjustments within the legend. It can be a simple statement. We don't need a full explanation, but just explain what you did. I recommend presenting results from one experiment. If it's this is your example of how the experiment worked, when you repeated it 5 times, you're just going to show one. You don't have to cut and paste multiple figures from all the best spans from out of your 5. Just show one that works the best. And before you go to submit the paper, compare the raw data with the final figure. Sit down with the raw data, label it, say this is going with your manuscript, save it in a special place so that you always have access to it, and take a look to make sure that what you're sharing with the reviewers is really still holds true to what was originally captured. And if you do receive a letter from me asking for the original captures, don't worry. If you have the raw data available, you send it to me within, usually people send it within 24 hours, and within 48 hours your paper's back in publication. All we want to do is make sure that what's shown is really what's originally captured. And by saying here it is, we just forgot to add a lane or add a line or space in between our splicing. It can go right back in. I'd rather make a correction than to do anything else. But if we don't have the raw data, or someone lost it, or someone left the lab and we can't find it, we went on vacation, there's biblical explanations, there was a fire, a flood, the computer crashed, then we can't publish the paper because we need to make sure that there's raw data available for every experiment. And so we don't often ask because we trust that there is raw data available, but if we have concerns, we'll ask for it, and we want to be sure that you want to be sure that you have immediate access to that raw data. Okay, so our guidelines are here on this website. And my last plug is that we have APS in collaboration with the Biomedical Engineering Society and the Society for Biological Engineers recently received a grant from the National Science Foundation to develop some ethics education materials for graduate students. We have developed the teaching modules and they're being final revised right now. And so if you work with graduate students or you're an RCR instructor and you'd like to have access to some of these materials on the topics of publication ethics, including authorship, conflict of interest, plagiarism, overlapping publications, data presentation, and research misconduct, just please let me know. We should have them available by this fall for anyone to use for free.